Hello friends, we are talking about developmental biology, and we have seen the introductory part about developmental biology and why we study developmental biology, and then we have seen the sequential step of embryogenesis, which is drawn here. That means the fusion of egg with sperm, then production of zygote, then the division of the zygote. Now cell division of the zygote, which you can call as cleavage, will give rise to mass of cells layered in different uh, or oriented or placed in different planes to make a three-dimensional cellular structure, which is called a blastula stage. Then these cells will start to mature, and as they mature they get the signal of producing different layers during the gastrulation step. Now this is the most important step above all, because at this stage those cells that we can derive from this blastula will be destined and committed to produce a special type of cells. For example, during these blastula events, the most important event is the production of different layer of cells. Now those different layer of cells will generate different type of organs and different type of organ system inside the organism's body. For example, during this gastrulation, it will develop into three different layers. One is the outer layer, which is called an ectoderm, and innermost layer is called the endoderm, and the layer which is placed middle of ecto and endoderm is called the mesoderm. So these three layers are generated. Now from those three different layer of cells, different type of tissues generated during the development or during the embryogenesis. Now a part we have talked about this whole picture is called the embryogenesis. Embryogenesis means it is a production and maturation of a fully functional embryo from one cell which was previously zygote. Okay, now we'll be studying this gastrula phase in detail in this particular video. So the process of the production of cells and production of these different cell layers during the embryogenesis at this stage will be called gastrulation. Okay, the phase is called gastrula, and the production of this type of cells from a chunk of one type of cells which are totipotent in nature, and they commit into a particular special type of cell via the process is called the gastrulation. Okay, now in this gastrulation, the most important event I've told before, that the production of or arrangement of these cells in different layers: outer layer, inner layer, and middle layer. So for this particular thing to happen, there are again sequential events, because remember, in developmental biology everything happens in sequential events. Okay, and timing is very much important, and we have seen in previous video that in this gastrulation, at the very beginning of gastrulation, at the early stage of gastrula, the cells are not yet committed to the production of a particular cell type or organ type. After the gastrula phase is going on, at the later stage of gastrulation, the cells are committed to do their job. Okay, so now here let us talk about gastrulation. So we'll first draw the structure of or from the blastula, the late blastula, then we come to the gastrula. Now actually the step is drawn here is not particularly blastula, because blastula stage is also important because it provides the structural integrity of the cell. It provides the 3D structure or a structure like a ball, a hollow ball, which is in turn converted into gastrula. So simply what we will be seeing is a hollow ball, but all the cells that are present there is destined to produce every type of cell inside our body. But once it is converted into gastrula, it produces three different layers. That is the difference during this process. Okay, so let us consider this. So let's consider this as a blastula. Now during this blastula stage, what we are having layers of cells arranged in different regions. Okay, for example, we need to draw this like that. Say this blue colored region is a type of cells. So the cell differentiation begins from the blastula phase. It will be completed after the end of gastrulation. Okay, and gastrulation means most of the part of this embryogenesis is done. So from the blastula phase we are having this. So what we are having a layer of cell. Now this is a blue layer of cell, so I'm just drawing it like that, so that you can find this as a blue layer. So it's a layer of cell. Then we are having this red region of cells. Okay, and then finally we are having this black part. Okay, okay, so we have something like that. Okay, now picture, look at the picture, and the color representation is very important in this case. Now this blue colored region cells are arranged. So it's a hollow ball. Why I'm telling it's a hollow ball? Because all these shaded regions are solid mass of cells. All of these shaded regions are solid mass of cells, and rest part, this blank region is blank. Nothing is there. So it's a hole actually. It's a vacuole like. It is called blastocele. Okay, and the whole thing is called a blastula and blastocyst, and this hole is called blastocele. Yes, this is called a blastocele. Okay, and rest of the part is there, and the rest of the part means this blue colored lining of cells. Remember, I've told you that the differentiation stage begins from the blastula and then it completed in this gastrula. So these blue colored things, blue colored cells here are ectodermal cells. Red colored region is mesodermal cells, and this black colored thing is endodermal cells. So we are having layers of cell in this particular stage. So if, from the beginning, in the blastula stage we are having this different type of cells: ectodermal, endodermal and mesodermal cells. Okay, but they are arranged just in a small amount like that. But during the gastrulation, what they does is that they start to arrange in such a way which is uh, giving rise to the organism. Okay, so these are simply mass of cells. Now the cells are arranged in a region and make a hollow blastocele at the middle. Okay, so this is the structure, but the cells will arrange in such a way, but they are differentiated in ectodermal, endodermal, mesodermal type of cells, but they are not yet layered. Okay, but after this gastrulation, what we can see, the cells will be layered in such a way that in this whole whole ball like structure, outermost will be the ectoderm, middle one is the endoderm, mesoderm, and the inner one is the endoderm. But in this case we haven't find these things. Just cells give rise to different type, but yet they are not arranged in layers. Okay, so in the gastrulation we'll be seeing the arrangement of these cells in layers. Okay, let us consider now. Now this is called a blastocyst, and it is having blastocele and blastula stage. Now what will happen? This cell will start to modify. How? Because the cell has, as you can see here in this endodermal region, there are some new arrangement of cells. Those cells are called bottle cells. Okay, or bottleneck cells. So these bottle cells here, so let me draw this here like that. So this is a joint where bottle cells start to, so some bottle cells, I don't know whether you're able to see it or not. This green colored cells are the bottle cells. Now these bottle cells are really important because they are called the guide cells. Okay, the chunk of cells, these are some bottle cells, not one, many bottle cells. Now why they are called bottle cells? Because they are so they are arranged in this like this orientation. So the apical region of the bottle cells are narrower, but the basal region of the bottle cells are broader. So that's why they're called, they're looking like a bottle, like a bottleneck, and bottom part is spreading. So it is called a bottle cells. Now these bottle cells are called guide cells because these cells guide other cells during this gastrulation. Because what we know in this gastrula, the most interesting event is the movement of cells and lining of the cells one after another. It's a sheet-like arrangement, and movement of cells will be required. Now what we will be seeing in the future events, the bottle cells will guide other type of cells like the cells of ectoderm, mesoderm and endoderm to move at this directions. This direction, it's a anti-clockwise direction. We'll be seeing it. Okay, so we'll be seeing this anti-clockwise direction of the cells, and then they will arrange it in layers. So our actual goal is to produce something like that. Let me draw this. So actual goal is a production of this thing. Ectoderm in the outside, mesoderm in the middle, and endoderm at the last. Okay, so this will be our arrangement. At the end we will end up with something like that. So after the gastrulation we will find something like that. At the middle we will having the hole which will make the gut, right? Now in this case it will be converted into this kind. How? Let us consider here. Okay, so here the bottle cell will guide other cells to move. Now as the bottle cells are formed, it will guide other cells to move in this direction. The cells will start moving, and as they are moving they start to make a small hole like structure, and this joint point of this bottle cells and the ectoderm and the mesodermal cells, it is forming a lip like structure, just like our lip. This opening, right? A small opening like structure. It is called the dorsal lip. So as they start to move, so let me draw it again here like this. So as they start to move, the movement will look like this. So as all the cells are spreading, and remember, so not only the cells are spreading, so if we need to spread the cells, we must have access to development of more cells. So cell division is going on in a rapid speed during this stage, and they are moving. So say here the cells are moving, and let us consider this like this way. Okay, and here are the bottle cells. Okay, something like that. Okay, and say okay, okay, so this will be the structure after some time. Okay, movement of cells is going on, so we can see the movement of cells are going on in this way. And sorry, one mistake is there. So say here it is, and okay, so this is a movement, and those bottle cells are placed there. So as they start migrating, the cells start migrating in this direction, anti-clockwise direction, and what we start to form a structure like that. So it will form a lip here. Now this region, the formation of lip is called dorsal lip. Okay, and as the cells start migrating, what they are producing here, they are producing a region which was previously blastocele, which is a large region, but now this blastocele is constricted because other cells are start migrating and filling the space. So it will lead up to the formation of a small region of blastocele. It is called, this particular region is called, so there must be this part, we must draw this. Okay, so this part is called then Archaneteron. So the blastocele is constricted due to the movement and movement of cells onto this region, and what they produce is called Archaneteron. This is again another hole, another type of vacuole, but this is now called Archaneteron, and it is also called primitive gut because this is the first type. So it's a very earlier type of gut of the organism, so it's called primitive gut, or also called Archaneteron, right? So let me write Archaneteron, or it is also called primitive gut. Okay, so you need to learn these terminologies because a lot of questions come in CSI and NET providing these terminologies. Okay, so the movement of cells is going on. So from this part what we can see, so these cell lines are moving, cell layers. So these mesodermal cell layers are moving here, endodermal moving, and the ectoderm is also moving. So this can eventually give us the idea is that, so this is the endodermal. Now the idea is giving us that this endodermal region is going to be inside, and rest of the regions, mesoderm will be the middle of the ectoderm and endoderm. So actually from this part we can see that if the cells continues to move at this direction, it will form a structure like this, right? And the middle is the gut or Archaneteron here. Okay, this is called dorsal lip, and as they are moving and this site is expanding, and it will form a blastocele and a blastopore like structure, what is called a blastopore, because this will form a porous region. It will be called a blastopore. From this region, those blastopore region will become anus of that particular organism. Now here we are discussing about the development and gametogenesis, not gametogenesis, sorry, embryogenesis of frog xenopus. So write it down. The example is given for frog xenopus. So they are moving. Now after the stage, what will happen here, what will we can see the movement of the cells. So after the movement, after few time after few days, the cells at this position again give rise to another type of bottle cells. So bottle cells previously formed at this particular location, now it will form the junction between the mesoderm and ectoderm of another region, because we are having the mesodermal ectodermal axis in two different points. Bottleneck generates at the region of mesodermal ectodermal region. It generates first here, now it will generate here. So from here we'll be seeing that bottle cells will generate. So another set of bottle cells will generate. Okay, so let me take here, bottle cells will generate here again. Now as bottle cells are generated, as we all know, bottle cells are guiding. The ability of bottle cell is to guide the cells, to form layers, right? So here they again start to guide this type of cells to go and form layer like structure. Okay, so what we can see, they start to form again a layer like structure. So as they are making invaginations like that and start to form the layer like structure, so they are moving here in this direction, and as they are moving in this direction, so eventually they are making the space for endoderm really shorter. Endoderm comes at the inside, and rest of the regions will be outside. So what will it look like? It looks like something like this. So again, say this is the beginning point. Here it was for the beginning, so try to follow this because it's very important. Now as they are moving in this direction, so they are moving here in this direction, and also the mesodermal region is also moving, so they end up with something formation like this, and the endodermal segment are constricted, so they will be here, like that. Okay, so at the end what we can form something like this. Okay, so at the end we will form something like this, because this bottle cells again guide the cells to go at this. So what will happen, again this lining of the cells happen and form something like that. Okay, so what we end up with here, we end up with three different layers. Outermost layer, which is a blue color layer, which is ectoderm. Middle layer, red color region, which is the mesoderm, and the inner black color region or endoderm. Okay, so after certain time this will fuse with one another and form something like that. Now in this particular place, the regions ectoderm, and this is a mesoderm, and again the endoderm. So remember, in case of the blastula or the late blastula, when we are having the blastocyst and blastocele and the formation of a hollow ball, we are also having the different layers, not layers, different type of cells, ectodermal, endodermal, mesodermal. But the important thing is that in those cases, we don't have the alignment, we don't have the arrangement of cells in this layer structure. But after the gastrulation, we end up with the layering of the cells, which is the most important part during the gastrulation. The layering of the cells, arrangement of the cells, and what it forms two different regions. Okay, it forms a region, and where we end up with a blastopore, and this will become the anus of the organism. And during the second bottle cells formation, it forms another lip. We call them ventral lip. Now formation of this dorsal lip and ventral lip are also important, because this dorsal lip and ventral lip will dictate whether which part is going to be the dorsal region and which part is going to be the ventral region of that organism. Okay, so end up with this kind of structure. Now this ectoderm, mesoderm and endoderm, the different layer of cells are having different functionalities. What are the functionalities? This type of cells will give rise to specific type of organ systems in the body of organisms. For example, the ectodermal cells will give rise to the skin cells and also sensory organs. Skin cells and also sensory organs. Mesodermal cells, which is in the red color here, they will give rise to, here this mesodermal cell will give rise to this mesodermal cell will give rise to reproductive system, bone system, okay, and also blood circulation system. But the endodermal region cells will generate, and this mesoderm also produce the reproductive system. The endodermal cells will produce digestive system and respiratory system. Okay, so the type of cells, so these cells, this ectodermal, mesodermal and endodermal cells are specialized cells. They will generate specific type of organ systems in the organism's body. Now this differentiation made in the late blastula phase, but the commitment of the cells is seen in the late gastrula phase, and the arrangement after the arrangement. So we can find the commitment of this type of cells to produce the specific type of organ system or cells of our organ system only after the late gastrula phase, when the arrangement of the cells happen in these three lines. Okay, so this three layer arrangement of the cells during the gastrulation is very important for the commitment of those cells to produce specific type of organ systems. Then when they produce specific type of organ system, combining them, we can have what is called an organism. Here in this case, xenopus frog, or it is a normal xenopus frog. Okay, now any problem during in this different stage, any problem during the phases of this gastrulation will lead to the malfunctioning or abnormal embryo, and this abnormal embryo will develop into an abnormal adult xenopus frog. Okay, now this is very important. Developmental biology, again I'm telling you, is a sequential steps, and timing is very important. Okay, now the stages of this gastrulation can be easily visualized and utilizing dyes, because if you stain those cells with different dye, we can track the movement of the dye during time, or during this development with time, and you can find something this. Okay, so these are the sequential steps of gastrulation, and I hope this will help you to understand. Thank you.